applications. The presenters are Google Chain. Let's welcome presenters with big hands.
Hello everyone, I'm Wu Guoqian and I'm here to talk about my topic, research on the effects DEHP has on the sperm of zebrafish. First, let's look at my motives. Three motives. The first one is the first PhD scandal which happened in Taiwan in May 2011. And the second one is the researches on DEHP impact on zebrafish fertility. The third one is the effects that DEHP has, which are these three. First of all, let's understand what DEHP is. DEHP is basically a thing that you add in your plastic to make it more flexible. And there are some effects that DEHP has. The first one is that it is an environmental hormone, meaning that it exists in the environment and it acts like a hormone. And particularly DHP is similar to estrogen. And estrogen is a feminine hormone, so if DHP is in your body, it may trick your body to think that it has too much estrogen. And also DHP can cause cancer, such as liver cancer. And lastly, DHP can induce male infertility, which basically means that you won't have babies. Now to my aims. I want to understand the effects DEHP has on the sperm of zebrafish. So I elaborated into three sub-aims. The first one is the activeness of the sperm, and the apoptosis and death of the sperm, and lastly, the sexual structure of the sperm. The independent variables I used in this research are DEHP concentrations and sperm exposure time. Now to my methods. First, let's look at sperm activeness. Activeness defined in this research is motility and linearity. Motility is how much the sperm moves, and linearity is how straight the sperm moves. And the tool I used for this particular experiment is called CASA, which is computer-assisted sperm analysis. It is basically an optical microscope and linked to a computer. CASA was originally designed to analyze human sperm. And as you can see in the right picture, these are zebrafish sperm. They are very small compared to human sperm. So I have to change some variables in order to observe zebrafish sperm. And now I have to extract my samples. Basically, I squeeze my zebrafish, and in the other hand, I hold a pipette to suck out the sperm. And after extracting it, I preserve my samples in an epimorph filled with PBS and water or DHP. And while doing each experiment, I dilute the sample because if you want to excite zebrafish sperm, a certain osmolality is needed. And each experiment has to be done in 30 seconds, or after 30 seconds, the activeness of the zebrafish sperm will decrease greatly. And this picture is me doing the experiment. Now to sperm apoptosis and death. Apoptosis, what is apoptosis? It's basically a programmed cell death. And the tool I use for this experiment is called a flow cytometry. First, I mark my samples with fluorescent antibodies. These antibodies attach to my samples, and under certain conditions, they will glow. After attaching the antibodies, I excite them with lasers of certain wavelengths. Then I make my samples pass through a small tube cell by cell, so that the computer can do the counting and then the plotting, which you can see in the upper right corners. The two antibodies I use are annexin-5. Annexin-5 clings to the cell membrane, and PI. PI clings to the cell interior. And how do I determine between apoptosis and cell death? If there is only annexin-5 positive, which you can see in the upper right corner of the page, there is only one antibody that is attached to the cell. This means that the cell is at an apoptosis stage, which is dying but not dead yet. If both the antibodies are positive, as you can see in the lower picture, that means the cell is dead, for the membrane is greatly damaged and the PI can get inside. And now, lastly, sperm structure. The tool I use for sperm structure is an electron microscope, which you can see in the lower right corner. And preparing the sample. Basically, I follow the protocol, but while first doing my experiments, I followed the original protocol. And I found out that most of my images are greatly distorted, or my samples are greatly damaged. And I did some research to find out the reason. The reason for that is osmolality, because while following the original protocol, I changed the osmolality so that the sperms are greatly damaged. So I did a small adjustment by adding PBS to sustain osmolality so that the sperm will be intact. And after preparing my sample, I put it inside a microscope and get my images and do the analysis. And you can see an example of an image. Now to my results. First of all, let's look at sperm activeness. After 72 hours of exposure to this concentration of DHP, the negative activeness of zebrafish sperm has greatly decreased, as you can see in this table here. And the blue numbers show that it decreased. And I can conclude that DHP does affect the sperm of zebrafish. Next, let's look at mitosis death. After 72 hours of exposure to this concentration of DHP, most zebrafish sperm are at the stage of cell death, not apoptosis, because apoptosis is usually observed in somatic cells. Somatic cells are cells that have 46 chromosomes, which sperms apparently do not. And also from these results, I can conclude that DHP decreases the activeness of sperm by inducing cell death. And the third part is sperm destruction. After 72 hours of exposure to this concentration of DHP, the cell membranes of zebrafish sperm are greatly distorted, as you can see in the bottom 4 pictures, compared to those not treated, which are the upper 4 pictures. And this further explains the high rate of sperm death. And now to my conclusion. I was able to use 3 different kinds of microscopes to observe the effects DHP has on the sperm of zebrafish. With the help of CASA, I concluded that DHP decreases the activeness of sperm. And by using a flow cytometer, I found the reason for that decrease, which is cell death. And lastly, through an electron microscope, I gathered even more evidence to support that conclusion. And with these findings, I can say that DHP decreases the fertility of zebrafish by inducing the death of the sperm, or in easier words, DHP kills the sperms of zebrafish. And because zebrafish is a monoorganism, this conclusion can suggest the impact DHP has on the human reproductive system.